Good afternoon, everybody. Welcome to STEM Inspiration Week. Your camera and your microphone should remain off, please, to minimise background noise and cause distractions. Please note this session will be recorded. Look at the hole on the faces of the presenters now. They're going to be there for posterity. Can you please use the link, the chat, to post any questions, which we'll pass on to the presenters. And I'd really like to welcome Cindy Smidt and her colleagues from Wellcome, and they're going to be talking about mosquitoes and how we're trying to deal with mosquitoes. I really hate mosquitoes, so this one's going to be really interesting for me. And how they're going to look at mosquitoes as a vector for transmission of malaria, and I presume some other horrible, nasty things that mosquitoes always give us. So I'm going to pass you over to Cindy. Cindy, can you take the screen share for me?
Share that one. Can you see?
I'm just checking. Yes, I can see that. Fine.
Lovely. So I'd first like to introduce my colleagues. So Francesca Gale, who is the Education and Learning Manager at Wellcome Genome Campus, and she'll be here to support and answer any questions that you have as well.
Yeah. And also just down here as well, we've got M. Dixon, who is our Education and Learning Officer at the campus as well. So we're here to answer any questions. And yeah, if anyone's got any questions about anything outside of malaria and genome research, we can help with that.
Thank you, Fran. Hello, my name is Cindy Smidt. I'm an Advanced Research Assistant at the Wellcome Genome Campus. I'm working with the malaria parasite life cycle. My main role is to look after them, our mosquitoes in the insectary, breeding and maintaining their colonies, as well as infecting them with rodent malaria. So first I'll tell you a bit about malaria. So this is our campus. It's very big. And we do our mosquito work somewhere in this building. I don't know specifically from that kind of view. So what is malaria? Malaria is a life-threatening disease which is transmitted to humans through the bites of infected female Anopheles mosquitoes. It is only the female mosquitoes that bite you. About 3.3 billion people, half of the world's population, are at risk of malaria. Causes of malaria is a single-celled parasite called Plasmodium. This is how they look. So these blue bits and the red bits are your red blood cells. There are more than 100 kinds of Plasmodium that can infect many animal species such as reptiles, birds, and mammals. They don't interlink as well. So one that affects reptiles cannot affect humans. Same with the rodents, birds. However, the primate malaria can affect humans as well. Plasmodium falciparum and Plasmodium vivax are the most common types of malarias that infect humans. This is the malaria that I work with, Plasmodium berghei. And as you can see, they're in the red blood cells. They're rupturing as well and floating around. So the Plasmodium life cycle. So you first get stage 1, transmission to humans. This is by sporozoites. When the mosquito bites your skin, in its saliva is sporozoites, which look like these. That's from a microscope as well, from dissections I've done. These little squiggly bits are sporozoites. They get injected into your bloodstream. They find your liver and they reproduce there. Then they rupture from your liver and infect more of your blood cells to reproduce, and then they make male and female gametocytes, which then gets taken up by the mosquito and that gets reproduced. So the gametocytes will mate, produce gametes, zygotes, and into oocysts. And once the oocyst, which is in the midgut of the mosquito, ruptures, it'll make its way to their salivary glands. But sometimes that process can kill the mosquito as well, depending on how infected it is. And this is currently where malaria is a problem. So some incident facts. 225 million cases of malaria worldwide, and there's 781,000 deaths every year. A child dies of malaria every 30 seconds. Over 90% of malaria deaths are in Africa. 200,000 newborns die each year as a result of malaria. So what next? Do we completely eliminate mosquitoes from the ecosystem? There's methods developed that can sterilize males, and has been proven effective that they can mate with sterile males can mate with the females, and then they don't have viable eggs, so it just ends there. But how would that affect the ecosystem? Other animals, plants, humans? We can modify parasites to make sterile parasites, gametocytes, so no male gametocytes, so that there is no production line as before. There's lots of anti-malaria drugs, and they're trying to find different drugs that can fight malaria because there's a lot of resistance. And that's part of what the project I'm on is focusing on, trying many different drugs and seeing if it can be used to hit the liver stage. So the project I'm on is that we want to stop the parasite getting into your bloodstream. So once the parasite ruptures from your liver, you get all the symptoms, so all the sweats and feeling very lethargic, and that's the part, all those symptoms that can really have a strain on your body. And if we can have a drug that affects the liver stage first, then you wouldn't get the symptoms, and it's possibly, hopefully, a much better survival rate. But we'll see how that goes. So let's take a look inside our mosquito lab. Okay. Can you see that one? No. Let me see. Stop share. Okay. So first we have to scan in to go into our inner lobby area. And this is our inner lobby. We have a cold air shower here when we leave, because it will have a negative pressure that pulls all of any mosquitoes that have decided to escape back into the insectary to make sure that nothing gets out of there. So if we do have any infected mosquitoes, they don't get into the environment, or they don't start breeding in this environment, although it's probably a bit cold for them, this species, which is Anopheles stephensi that we mainly use, and they're from India. And especially currently, it's a lot colder than that. All right, so then we have to wash our hands and put on our PPE. So we've got disposable gowns that we need to make sure that we zip up all the way. And as you can see behind me are incubators as well. Nice fancy ones from Germany. Then we've got some safety specs. And since I've got glasses, I've got to put them over my glasses. And we've got gloves. All right, so this is a walkthrough of our entire insectary. So first we have the Bergy lab area where we have all of our microscopes, dissection scopes, fluorescent scopes. That incubator doesn't work right now. Mosferatu and Mozambique. So we've named them so that we know if there's different species in it. So these are currently my infected mosquitoes. They're kept at 19 degrees Celsius and 75% humidity. The infection doesn't like it too warm, so it's better to keep them at a nice cool 19 degrees. Then we move onwards to our breeding room, which we have to have separate doors because we don't want the adults getting into this room because there's lots of water, and we don't want the females to bite us and then lay some eggs randomly around the room. So lots and lots of larvae pans. Then we go through to our CL3 area, which we're currently not using, but if we were to do any Plasmodium falciparum human malaria infections, this is where we would do it in a very secure area. This is me trying to kill a mosquito. because it escaped. The best way is to catch it like that or spray it with ethanol, as you see. Have to spray with ethanol before putting my hand inside the cages, which these ones were blood fed. So we blood feed them on a Friday. We put the eggs egg bowls in on a Monday, collect the egg bowls on a Tuesday. We actually feed them fish food, so cichlids sticks, liver powder, and liquefy liquid. And so when you have a pond in your back garden and you start feeding your fish, the mosquitoes are actually loving it as well. So it's great breeding ground for them. So I always have a hot water tap running, just in case there's anything that wants to escape that way. Boiling water, that's at about 70 degrees Celsius, will kill them. But obviously you can't do that to your pond because you've got fish swimming in there. So then we use some RO water because they don't generally like tap water. They do live fine in it. We have accidentally made that mistake before. They're perfectly happy in anything. The dirtier water, the better usually. But we use RO to try and make sure that they don't have any infections and we keep them a nice, clean, healthy colony. So now I have to try and catch the adults that have stayed in the egg bowl. So a lot of them will drown themselves as well in it. So in order to fight infection as well, we do a bleaching of the eggs. It's a 1% solution. And this doesn't kill the eggs or the mosquitoes. They hatch fine. It's just to take anything that's from the outside of the egg and kill that. There can be bacteria attached to the outside of the eggs from the adult mosquitoes being in the egg bowls. So that's why when we have to remove the adults from there, and we have to bleach the eggs as well. Our bins are also autoclaved so that all infection is gone as well on there. And we keep everything nice and clean. So now to catch some adults. So the little black dots that you'll see inside, all these bits here are the eggs. And the eggs like to stick to each other so that they don't float off. And they usually stick to everything that's on the side as well, so that they can stay where their parents laid them. And because a lot of the adults do like to drown themselves in the egg bowl, the eggs stick to them. So in order to get them off, we have to tap them on the side. So you can see some of the eggs coming off as I'm wiping the dead adult mosquito along the side. Yeah. It is a long process as well, usually, because as you see, I've got 4 egg bowls. And I've got to do this to every one. And how I kill them is it's an azo wipe, which is an ethanol wipe. And I put it in there and squeeze them. So crushing them as well as kind of disinfecting them in a way. Now this is how we bleach our eggs. So we've got a vacuum pump and a SteriCup filter. So I'll put the eggs in the top section, as you see. I'm going to attach the pump first. And carefully pick up the cone, pour the eggs in. That's filter paper that we use as well to keep it nice and moist, because if the eggs dry up, they're no longer viable either. And I usually get them stuck to my gloves as well. And that's RO water as well in the squirty bottle. So adding my bleach dilution. Putting on the pump, and you can see the water trickling down. So it's being pulled through the filter to the bottom. So the eggs won't go through this filter here, just the water. Then once we've done a bleach run through, we do another 2 lots of RO water to make sure that the bleach is washed off. Okay. Once the eggs are have hatched, which usually take about 2 days, we have to separate them into different larvae pans. So we add the usual food that we did before into the egg pans, add some RO water, and then we add about 150 to 200 larvae. And then we have to cover it with netting, because in case they do emerge into adults, and we don't want that going in the room. So once they've developed into pupae, we have to collect them. See, there's lots of pupae and different stages in there, so mainly larvae, pupae, and some eggs that haven't hatched as well. And we clean it with boiling water, because we can't clean it with anything else like bleach or Virkon or anything like that, because that could kill the mosquitoes, as well as kill the infection that we are trying to do in the future. I like to put them all into one pan. As you saw from the walkthrough, there is so many pans that I have to try and empty out. So I put them all together. Then they go into this little jug. As you can see them moving around. And they have to go into these glass beakers. Have to have only a single layer of them, because they make it stink like dirty pond water. They are very dirty buggers. See all of them moving around and wriggling. Just a nice single layer. So I usually put about 4 of these per large cage as well, and then add some more water so that they can swim around a bit and be happy. That's our fructose in there. That's what the adults feed on generally, with our filter paper that I roll into a nice cone. And put that in, and then fold over the top, because mosquitoes aren't the brightest, so they will drown themselves in there trying to get to the fructose, and they won't know how to get back out. So we have to make sure that it's nice and closed off. So currently our colony cages are being kept in the CL3 area because the incubator is not functioning. But thankfully there's no human malaria work going on, so there won't be any risk. So these are the adult cages that we use. And it's full of netting, and they can't actually escape from that. There's also a bit of blue tissue at the bottom, because mosquitoes do actually make a lot of mess, especially when they defecate. So when they're pooing, they make so much mess, as you can probably see in these other cages on the right. And then dead mosquitoes also as well in there. And then fructose in, got to tie knots in the cage so that they can't get out. They definitely can't untie a knot, thankfully, I have checked. So that's good. This is how we select females. So it's only the females that we infect, because they're the only ones that drink your blood. And they are attracted to heat and CO2. So if you starve them for 24 hours and then put a very hot jug in the middle then you can see that they all flock to it and they think that they're feeding, and you can sometimes see them putting their mouth parts, their proboscis through, and that can go a real distance because obviously it needs to get through your skin. And this is our vacuum, so it's just a bug hoover, a kid's toy. And once they're in there, put them into a new cage, blow them out of there, and then it's a cage full of just females. So we can monitor how many of them get infected, rather than just seeing lots of boys around. And this is how we usually do our blood feeds. So this is a very hot glass beaker. It's blood warmed. It's blood plus serum mixed together with parafilm over the top, which is to replicate skin so they can pierce through. And it's on top of their cages, upside down with a hot, to keep the blood warm. Then we have to also blow into the cage so that they're enticed to think that it's feeding time. That's an animal that they're feeding from. And here's some blood-fed females with nice fat bellies. Sometimes they overfeed, so they get very greedy. So when they get very greedy and feed too much, they unfortunately die because they're just too full. So as you can see, I've got lots of casualties here because they just fed too much. Right, and that is that is within our Mosquito Lab. And I'll just go back to the presentation. Right. So that's inside of our lab, and from we have to work with different life stages. So I was going to ask everyone if they knew what stages these are, but unfortunately that got revealed to this one. So these are the eggs. As you can see that they're very, very small and oddly shaped, but that's because they tend to hug onto each other and get stuck to each other. And they usually make a nice raft on top of the water for that. Can't get this to work. Oh, geez, one moment. There we go. Right. So sorry about all that. So the mosquitoes' life stages goes in 4 stages. So it goes from eggs to larvae to pupae to adult. So you saw the eggs. This is the larvae stage. So they have their mouth parts here and the tail end here, and lots of little sort of like legs, but it doesn't help them swim that much. It's mainly just their long body that wriggles. And they do look very different to the UK species and to a lot of other species, but I've seen some in my pond at home, and they're a lot bigger. These ones are tiny in India. And here's our little pupae, which you might recognize the design from a very famous film. I don't know if that's where they got their inspiration, but it looks pretty much like them, little aliens. And this is the stage that they go through before they become adults. Now let's see if you can identify. Question for you, which one is the male and which one is the female? I can't seem to get the chats up.
If everybody can write in the chat function to say top or bottom, which you think it is, so top whatever you think it is and bottom whatever you think it is, that will help Cindy. Thank you.
And I can't see the chats at the moment. It doesn't seem to want to come up while I'm in this view.
I'll just read out a few of the answers. Some of them are saying top right, bottom female, top female, bottom is male, top. I don't know whether that's female, top female. Are they? Seems to be coming up that the top one seems to be female. It's very leggy, someone's put.
It's with how they're laying down. So they're very hard to place. So once I've placed them onto a slide, they're a bit different and flat. So currently they're being suspended in ethanol so that they are not alive, but they do still move because they're in liquid. So the top one is female and the bottom is male. Well done for everyone guessing that. So the reason that there's a difference is, the reason you can see the difference is that their mouth parts. So you can see with the female, it looks kind of like a needle because she's got to pierce your skin and she's the only one that blood feeds. Whereas the males, they only have this kind of filter mouth part because they're feeding on sugars. So they can't actually pierce anything. And the females also have a much larger abdomen than the males, which you can't see very well from this angle though. And then this is the whole life stage. So imagine this is your pond at home or when you go out for a walk. You get your adult female mosquito that's had a nice blood feed off of you perhaps, and then goes and lays its eggs in a little raft, which different species of mosquitoes' eggs do very different things and look very differently. Then once the eggs hatch, they become larvae and then they become pupae. So for ours, it takes about 2 days for them to hatch. Then it takes, then they're larvae, and then it takes about 5 to 7 days for them to become pupae. And then after that, it's a day or two when they become adults. So we have to quickly move so that we don't have adults in the room, so that we don't get bitten. And thank you very much for listening. I hope you enjoyed it, and special thanks to Francesca and STEM team for all their help. And any questions? And now I can see the chats.
Thank you, Cindy. Just going through the chat, we're just waiting for a few people to add some questions in the chat function. So if there's any questions that anybody's got, you've probably covered everything. That was a great presentation. Thank you. How long do they live for?
That varies on the species and varies whether they're in captivity or out in the wild. So I've had in our lab, they don't usually live more than 4 weeks, but that's by our choice. I have starved them for about 2 weeks before, and they do live fine without food for a while. So it just depends on the species and depends what their food source as well. It can vary very greatly. And the females usually live a lot longer than the males.
So we have a question. Do some mosquitoes like specific blood types?
It's more, I believe, to do with the CO2, so what you emit. And if you emit a bit more CO2 than another person, they're more attracted to you, as well as your, if you're quite a warm-blooded person, they get more attracted to that. They are just feeling your CO2. So it's not specifically to your blood type, it's just whether they can smell you better than the next person.
Okay, I don't know if you can see the chat. We've got a student that's asked whether mosquitoes can spread HIV.
I'm not sure about that one, actually. Fran, do you know at all? It is a bloodborne.
I've not heard of anything overly documented, but in theory, I suppose, because it's transferring blood of from one person to, well, but that I think it's unlikely, but possibly not impossible. I wouldn't say it's commonly known that it does that, but again, we can find that out for you if you want to know that. And if anyone has got any questions that we can't answer right now, you can email us at engage@wgc.org.uk, and we will find out the answer to the questions that we can't answer just right now.
And here's a good one. How many species of mosquito are there? I think it's a lot, I'd imagine.
Thousands upon thousands. So they're part of the order Diptera, which is, there's 3,000 insects in that, and there's so many different species. Not all of those 3,000 are mosquitoes, but there are thousands of different species.
And someone's asked, what different colours can they be?
I'm not sure. It depends on the species, because we've got two different species in the lab, which is Anopheles ngoso and Anopheles defensi. Just from looking at them in their cages, they don't look any different. But I wouldn't know that, sorry. But we can probably find that out as well. No?
No, that's the blood used in the lab in the upside down beaker. Where does it come from?
That comes from the NHS. So when you donate blood, and if they have a surplus, so for instance, my blood type is O positive, and that is the blood that we actually get, and that's one of the most common blood types. So because the NHS has surplus, say, of that one, or it doesn't fit their criteria, then we get that.
So I've just had a really good question, which is, why do male mosquitoes not bite?
Because of their mouth parts. So the mouth parts are very different, as you saw in the pictures. But it's mainly also because the females need the blood to produce the eggs and then to lay them.
What country are they mostly found?
All over the world, apart from the icy cold ones, I believe.
Oh, here you go. What do you mean by keeping them alive by choice?
Depends on how long we need them for. So currently with the projects of Plasmodium burgii infections, we only need them for a certain time frame. Colony maintenance, for instance, if we keep feeding the same cage, the females produce less eggs. So we don't feed them more than once for colony maintenance, and for infection, we only infect them once as well. So once they've done that, we no longer need them, so they don't live more than 4 weeks.
So we've got a question that two questions that I can bundle into one question. So, which is, what career academic pathway did you follow to get your current job, and why are you interested in studying mosquitoes?
So career academic pathway, to get the current job, I went through a BTEC animal management diploma, which got me into becoming an animal technician, which I enjoyed that. And I also studied zoology as a BSc honors degree. But the main thing that got me into this job that I've got is my experience as an animal technician, being organized. I think that is the main gist of it, just being very organized, because the project is run strictly from a timeline from infection, then 3 days later you have to passage the blood on. You can't do it a day after, a day before. Mosquitoes get dissected at day 21 because of the sporozoite development. So the main path that you need to do is, if you can be very organized and any sort of science subject that you're interested in. I know one of my colleagues studied parasitology, and she's doing that at the moment as well. And a lot of my colleagues that are mainly focusing on culturing Plasmodium falciparum do those sort of pathways.
Here's a really good one. In fact, there's two questions. They're basically very similar, which is, can mosquitoes get any diseases from humans?
Depends on the diseases you mean. It's not really like the common cold or COVID, for instance. We can't pass that on to them. I think it's only the vectors that is used, the diseases that uses mosquitoes as a vector, but that is something we can also find more out about if you'd like some more information about that. The main one I know of is malaria and a lot of bloodborne diseases.
What's the favourite part of your job?
Is it okay to say killing them?
Yeah,
Because
yeah.
I do get bitten quite a bit when I'm not very careful, and they do fly around and I'm not paying attention and then they bite me. That's one of the favorite satisfying parts, that if they bite me, I can kill them.
You're one of my favourite people. Just for that, I really hate them. And actually, this is a really good one because I think I know the answer to this one. Can humans be immune to malaria?
Would you like to answer that one?
I might be completely wrong, but I think sickle cell is a way of getting it effectively, which is a mutation against malaria, isn't it?
It's not something I know. You probably know more about it than me.
Oh, yeah. So,
well, I can jump in if you want.
Yeah.
So
Yeah, please do.
it's really interesting because malaria has been around thousands of years, and actually it's almost shaping the genome of humans by it's this constant race between parasite and human trying to outwit the other. And sickle cell anemia is one of those really good examples of how human blood cells have slightly changed, and because of the sickle cell, a bit like a crescent moon shape, the parasite can't get in. But there is a downside to having sickle cell anemia. It makes you very poorly. So if you do have that, then generally you are less likely to get malaria. And then you have the people who are carriers but don't actually have sickle cell anemia, and they are possibly a little less susceptible to it, but it's not to say they're completely immune to it. But that is one really good example and a really great question to show how we are constantly adapting to prevent ourselves being infected by parasites like malaria.
that was a good question. A really good question.
Yeah.
Can you get malaria in the UK?
Not currently, because it's been ridden. The drugs were very effective, and they did get completely rid of malaria here. But that depends if it can come back if the mosquitoes become completely resistant to all of the drugs that we use, and they can be transported in many different ways, sea, land, on you, etc. But currently, no.
I'll jump in as well. So actually about 100 years ago, it was in the UK, particularly in the wetlands of the sort of fenlands of Cambridge, but because of the way that we don't have as much standing water as well, and a lot of drainage and better living conditions, that's why it's not really in the UK. You can catch it if you go to a malaria endemic country and bring it back with you. And that's why people will always say, take your meds. Even when you come back to the UK, keep taking your medication, because you have to take it for about 12, 14 days after you've left the country, because if you got bitten on your last day, it'll take up to 14 days to get into your bloodstream, and that's when you start seeing the symptoms. So that's often when you've heard about celebrities like Cheryl Cole and Didier Drogba having malaria, it's because they've been to a malaria endemic country and perhaps not taken their meds as long as they should have done, and that's when they've had it in the UK and had to go to hospital.
And actually, there's a really good question that follows on from that, which is what happens if you catch malaria in the UK? So I think they're asking what sort of treatment regime you get.
That's the same. You would get the anti-malarial drugs in this country, and your doctor can prescribe them for you. And as long as you stick to them, you'll be fine, generally.
Do mosquitoes flock to different areas?
It depends on if there's a lot of food available for them, and activity, and it needs to be nice and warm, and water as well available for them to be able to reproduce. So it depends on the areas. So they will flock definitely to your pond. So if you have that in your back garden, maybe don't go out at dusk and dawn.
Here we go.
Mosquitoes can't bite each other because it's only they just want to feed for the sake of reproducing. So that's why they'll bite any sort of host just to get the blood from you, which they wouldn't get from another mosquito.
There's a good one there as well about can malaria mutate. Do you want to take that, Cindy, or shall I?
By all means, Fran.
Yeah, so the plasmodium parasite, it can mutate. So there's certain elements in certain genes. So when we say it's become drug resistant, it's because there's been certain changes to its DNA that now means it doesn't respond to the drugs. And this is a problem because if those parasites are able to spread, then that anti-malarial resistance will spread across the country. And there's already some signs that the frontline drug, which is called artemisinin, there's already a lot of resistance in Southeast Asia and spreading. So yes, yeah, it can mutate, and the implications of that can be quite serious, especially if the mutation means it's resistant to drugs.
Which is why we're constantly doing more studies on different drugs that can affect it, and whether we can attack the malaria parasite at different stages so that it might not become resistant because it hasn't got to another point of its stage.
You know, is it possible for malaria to be resistant to all drugs?
It is possible in a sense, but because there are so many different ones as shown on the slide, it is a bit difficult for all of the different malaria parasites to become resistant to all of it. It's also different areas that might become drug resistant to a specific drug, but other areas will be fine for that drug. So it depends if the parasite has mutated in a specific area and if that is carried to a different area by the mosquito. And then there is always a combination of different drugs as well that they recommend, so that even if the parasite becomes resistant to one drug, it gets another one that will kill it. So combination is usually effective.
So we need to wrap up now, but I've just got one last question for you. What happens to mosquitoes during the lockdown?
Well, I kept them going. They were perfectly fine in my insectary. They were ticking by, and they had no issues, although they did get a bit skinny, should I say, because they weren't being fed as often, and they weren't being treated with as much TLC as I usually do. But now they're nice, plump, as you saw with the pictures. They're enjoying their feeds as much as a mosquito can probably enjoy a feed. So they didn't have much of an issue. And if there's any more questions, please don't hesitate to email us.
Yes, we're wrapping up now. So thank you everybody for coming today, and thank you to Cindy and her colleagues for answering all the questions. I'm sure everybody learned loads of new things. I did. I always love sessions like this. I learned loads of stuff, even though I still hate mosquitoes.
Can I just add thank you again to everyone attending. I've just added a Google form link for you to give us feedback on this session. That'd be really great. We don't ask for any details, etc. Just as if you can get as much information from you about today's session, that would be fantastic. So thank you once again for joining.
Thank you everyone for joining and listening to me chatter on about my mosquitoes.
So if we can ask the presenters to stay on and ask everybody if they'd like to leave the call now, that would be fantastic. And we'll hope to see some of you at a later stage this week.
I always think some of them have fallen asleep. And they
I had so many in there because I think a lot of the teachers had shared their link with a lot of the students as well. And this was the biggest session we had register. So I don't know, they don't all do the right amount.
It's a lot more interactive than it was the previous session.